Mute and Nisha start.
Initially, the excretory ducts of both systems enter a common cavity, that is cloaca. And the urinary system develops from nephrogenic cord and the genital system from the gonadal ridge. So here is the diagram of the early embryo. As all of us know, in the beginning, during the embryo development, there is a trilaminar disc, germ disc formation where we have ectoderm, mesoderm, and endoderm. And later on, the part of the ectoderm pinches off and gives rise to the notochord, as we can see in this diagram also. I'll move the cursor. Yeah, this is the notochord which pinches off from this ectoderm. And later on, there is a craniocaudal fold in the embryo and lateral fold. So embryo assumes this sort of shape. So, and the mesoderm is divided into three parts, that is paraxial mesoderm, which we have studied in our anatomy, which gives rise to somites, that skeletal muscles, all those things, and the intermediate mesoderm here, and the lateral plate mesoderm, which is divided again into two parts, that is somatic mesoderm and splanchnic mesoderm. Splanchnic mesoderm giving rise to the lining of the viscera and somatic to the body wall. Fine. So today our main concern is about the intermediate mesoderm, from where the genital and the urinary system are going to develop. In this diagram, also mesoderm here, and the gonadal ridge will also develop here, and they will give rise to the genitourinary system.
Nisha, if I can interrupt you, kindly explain the right one. That is the most important thing. The
Ma'am,
right
it's
diagram, you
explained
have
in
to
the
explain
further.
that the intermediate mesoderm, how it is dividing into two parts, which is part of the kidney and upper part of the kidney,
Yes,
not
ma'am.
the lower
That's what,
part.
ma'am, in the previous slide I have explained. The nephrogenic cord will develop, which will give rise to the kidney system, and the genital ridge, which will give rise to the gonadal system. In the further future diagrams, it's been shown more clear. I'll explain them. I'm like, so in this diagram, we can see this is our gut, which develops from the endoderm, dorsal mesentery, gonadal ridge, and here the mesonephric duct and paramesonephric duct. So from mesonephric duct, most of the which will participate in the formation of the kidney system. So just giving the brief in the beginning, uh, the development of the kidney is from the metanephrogenic blastema and ureteric bud. So these terms will be used in the future slides. So I'm just giving a brief of that. The ureter will develop from the mesonephric duct, the urinary bladder except the trigone area from the urogenital sinus, and the trigrone from the mesonephric duct, the anterior part of the urethra from the genital tubercle, and the posterior part from the urogenital sinus. So I'll explain all this, how they are going to develop. So in this diagram, we can see that this is the nephro, pink one. This is the nephrogenic cord. So it is divided into pronephros, mesonephros, and metanephros. Pronephros is the very early kidney system, and it will be rudimentary later in life, and it will be functioning from the beginning of the fourth week, and later it is replaced by the it will degenerate, and it will replaced by the mesonephros, and later by the metanephros. Mesonephros will degenerate, and the mesonephric duct will be remaining. Metanephros doesn't have its own duct. It will use the duct of the mesonephric duct only will be used. And from the mesonephric duct caudal end, just before joining the cloaca, it will give a ureteric bud from where the collecting system will develop, and the metanephrogenic blastema, the metanephros, which will give rise to the excretory component. So we'll discuss all these in the further slides. And as everyone can see, this is cloaca, allantois, or which will later form the urachus, and the median, the remnant of it will form the median umbilical ligament and vitelline duct, no doubt. Yeah. So the development of kidney and ureter, there are three slightly overlapping kidney systems are formed in a craniocaudal sequence during intrauterine life in humans. The pronephros, mesonephros, and metanephros. Pronephros is rudimentary and non-functional. Mesonephros functions for a short period of time, like from the late fourth week, and it functions for 4 weeks, and then it is taken over by metanephros, which forms the permanent and which starts its definitive functions by around 9 to 10th week. So pronephros, these bilateral structures appear early in the fourth week. They are represented by a few cell clusters and tubular structures in the neck region. The pronephric ducts run caudally and open into the cloaca. The pronephros soon degenerate. However, most parts of the pronephric ducts persist and are used by the next set of kidneys. Now coming to the mesonephros, these large elongated excretory organs appear late in the fourth week, caudal to the pronephros. The mesonephros are well developed and function as interim kidneys for approximately 4 weeks until the permanent kidneys develop and function. The mesonephric kidneys consist of glomeruli, almost 10 to 50 per kidney, and tubules. The mesonephric tubules open into bilateral mesonephric ducts which open into the cloaca. The mesonephros degenerate toward the end of the first trimester and replaced by metanephros. Metanephros, the primordia of the permanent kidneys, begin to develop in the fifth week and become functional approximately 4 weeks later. By the time mesonephros perform their function, the permanent kidney develop from two sources. As I have already shown in the diagram, from the caudal end of the mesonephric duct just before joining the cloaca, the mesonephric duct gives rise to ureteric bud and or metanephric diverticulum. So it helps participates in the formation of permanent kidney, and the metanephrogenic blastema, which is pierced by penetrated by the ureteric bud and gives rise to the excretory component metanephric mass of mesenchyme. So coming to the definitive kidney, developing from two sources, metanephric mesoderm and ureteric bud. The metanephric mesoderm forming the excretory component and the ureteric bud forming the collecting system. So nephrons are formed until birth, at which time there are approximately 1 million in number in each kidney. So after that, no more nephrons are formed. Urine production begins early in gestation, soon after the birth, which start to form by the 10th week. Yeah. At birth, the kidneys have a lobulated appearance, but the lobulation disappears during infancy as a result of further growth of the nephrons, as well as increase in the interstitial tissue. Uh, Whereas there is no increase in the number of nephrons. So in this diagram, we can see here the blue component is denoting the excretory component and the yellow in the collecting system. So here we can see these are the from the metanephrogenic blastema. These are metanephric tissue caps. So what will happen? So in this diagram, as we can see, this is the collecting duct and then collecting tubules are there. So at the end of each collecting tubule, there is formation of these metanephric tissue caps, which will cluster and form the renal vesicles, metanephric vesicles, which will acquire a S-shaped uh, curve, and then from the proximal end of the S-shaped, here the glomerulus and Bowman's capsule is formed, like it is penetrated by the capillaries, glomerular capillaries, and forms the glomerulus and Bowman's capsule. The later part will elongate, differentiate, form the proximal convoluted tubule, loop of Henle distal convoluted tubule, which will join the part formed by the ureteric bud, that is the collecting system, and it will open into the collecting tubules, giving rise to the complete functioning nephron. So here we can see the formation of the both excretory component and the collecting component of the nephron. Coming to the collecting ducts of the permanent kidney develop from the ureteric bud. The ureteric bud is a diverticulum from the mesonephric duct near its entrance into the cloaca. The metonephrogenic blastema is derived from the caudal part of the nephrogenic cord. So we have seen in the previous diagram the nephrogenic cord. So the metonephrogenic blastema is derived from the caudal part of it. The proximal parts, as we have already seen, gives rise to pronephros, mesonephros. As the ureteric bud elongates, it penetrates the metanephrogenic blastema, a metanephric mass of mesenchyme, and each bud forms two new buds while penetrating the metanephric. So, in the previous diagram, also we have seen there is a kind of like each ureteric bud, while dividing, gives rise to two branches. So, there is a dichotomous kind of division. So, it divides around 12 to or more generation of division are there, forming the tubules. Position, blood supply, and direction of hilum. What are the changes in shape of the kidney? Initially, the infantile kidney is lobulated in appearance. It's growing, it's lobulated in appearance, and later the lobulations disappear. It requires the adult form, that is the smooth curvature of the kidney, and with the growing tubules and the surrounding mesenchyme. As we have already seen, but there won't be any further increase in the number of nephrons. The number of nephrons is fixed at birth. And the changes in the position of the kidney. When the kidneys initially develop, they develop in the pelvic region. But later on, they migrate to the lumbar region. Why so happens is there is a diminution in the curvature of the embryo, and that is one reason. Secondly, there are multiple organ systems. The pelvic is a very narrow cavity, and in that multiple organ systems, the urinary bladder, the rectum, the all the blood vessels, all the major components will be developing. So they will push the kidney and help in the ascent of the kidney from the pelvic region to the lumbar region. So there will be changes in the position of the kidney from the pelvic region to the lumbar region. Changes in the blood supply. When there will be a pelvic organ, they'll be receiving their blood supply from the more distal caudal branches of the aorta and from the common iliac arteries. As they ascend up, they receive branches from the more proximal parts of the aorta, and later on the renal artery will be formed. And the direction of the hilum. So in the early period of development, the hilum of the kidney faces anteriorly, but in the with the advancing gestation, there is a 90-degree anteromedial rotation of the kidney, which results in the direction of the hilum will be facing, which was facing anteriorly before, will now face medially. Sometimes there can be a malrotation, and instead of facing anteromedially, it may face outwards. So that will gives rise to a malrotated kidney. So in the diagram also we can see, yeah, the diagram we can see that here the kidneys are developing the pelvic region. They will ascend up to acquire their normal position in the lumbar region. The changes in the blood supply of the kidney from the further ascending parts, and yeah, I'll discuss it in the further slides also. The positional changes in the kidney. Initially, the primordial permanent kidneys lie close to each other in the pelvis, later shifts to a more cranial position in the abdomen. This ascent of the kidney is caused by diminution of body curvature and by growth of the body in the lumbar and sacral regions. They attain their adult position by the ninth week, contact with the suprarenal glands. Initially, the hilum of each kidney, where blood vessels, the ureter, and nerves enter and leave, face ventrally. However, as the kidneys relocate, they rotate medially almost 90 degrees. By the ninth week, the hila are directed anteromedially. Eventually, the kidneys become retroperitoneal, external to the peritoneum on the posterior abdominal wall. The changes in the blood supply of kidneys. During its ascent to the abdominal level, it is vascularized by arteries that originate from the aorta at continuously higher levels. Initially, the renal arteries are branches of the common iliac arteries. Later, the kidneys receive their blood supply from the distal end of the aorta. When they are located at a higher level, they receive new branches from the aorta. Normally, the caudal branches of the vessels undergo involution and disappear. The kidneys receive their most cranial arterial branches from the abdominal aorta. These branches become the permanent renal arteries. So, in case the caudal branches doesn't undergo involution and disappear, what will happen? So, we can see in this diagram, for example, this branch is persisting. It has not involuted, it has not disappeared. So, it's going to have compressed on the ureter and may lead to the ureter and hydronephrosis. Now, coming to the congenital anomalies of kidneys and ureters, under this heading, we are going to see the following anomalies: renal agenesis, malrotated kidney, ectopic kidney, horseshoe kidney, supernumerary kidney, cystic kidney disease, ectopic ureter, and duplicated ureter. We are going to deal each one by one. So, coming to the renal agenesis is the absence of one or both of the sides of the kidneys. That is, it can be unilateral or bilateral. Renal agenesis results when the ureteric buds do not develop, or there is a failure of the ureteric bud to penetrate the metanephrogenic blastema. When the ureteric bud develops, it penetrates the metanephrogenic blastema. When it fails to do so, even the metanephrogenic blastema fails to develop in the uh, metanephric tissue. So that results in failure of kidney developments. So both the events are essential. Separately, both of them can't function. The ureteric bed has to penetrate the metanephrogenic blastema to induce its differentiation into the kidney tissue. So in the unilateral renal agenesis, males are affected more often than females, and the left kidney is usually the one that is absent. Unilateral renal agenesis often causes no symptoms and is usually not discovered during infancy because the other side kidney usually undergoes compensatory hypertrophy and performs the function of the missing kidney. Unilateral renal agenesis should be suspected in infants with a single umbilical artery. In bilateral renal agenesis is associated with oligohydramnios and because little or no urine is excreted from the amniotic cavity, and this condition is incompatible with a postnatal life. Infants will with bilateral renal agenesis usually die shortly after birth. Now, coming to the malrotated kidney, if a kidney fails to rotate, the hilum faces anteriorly, that is, the fetal kidney retains its embryonic position, and abnormal rotation of the kidneys is often associated with the ectopic kidneys. So, in the diagram, we can see here the hilum is facing anteriorly. It has failed to rotate anteromedially, and in this diagram, we can see that there is an abnormal rotation in the kidney. So coming to the ectopic kidneys, one or both the kidneys may be in abnormal position. Most ectopic kidneys are located in the pelvis, but some lie in the inferior part of the abdomen. Pelvic kidneys and other forms of ectopia result from failure of the kidneys to ascend. Horseshoe-shaped kidneys. The poles of the kidneys, as we have seen, when they will be in the pelvis, the kidneys are very close to each other. The inferior parts, the inferior poles of the kidneys are very close to each other. Sometimes they are fused in the inferior region and gives rise to the horseshoe kidneys. The poles of the kidneys are fused. Usually, it is the inferior poles that are fused. The large U-shaped kidney usually lies in the pubic region. Normal ascent of the fused kidney is prevented because they are held down by the root of the inferior mesenteric artery. From the aorta, inferior mesenteric artery will be taking its origin. As the kidney is horseshoe shaped, its ascent is prevented by the root of the inferior mesenteric artery. A horseshoe kidney usually produces no symptoms because its collecting system develops normally and the ureters enter the bladder. It functions normally, and this condition is compatible with life. Approximately 7% of persons with Turner syndrome have horseshoe-shaped kidneys. Now coming to the supernumerary kidney, a supernumerary kidney with its own ureter, which is rare, probably results from the formation of two ureteric buds. For example, two ureteric buds are developing These are two ureteric buds. They will separately penetrate the metanephrogenic blastema, giving rise to the supernumerary kidneys. Here in this diagram, we can see the supernumerary kidneys. In this diagram, we can see the two ureteric buds developing. Now coming to the cystic kidney disease. The cystic kidney disease, kidneys contain small cysts which result in renal insufficiency. Cysts are seen and they range in size from a few millimeters to many centimeters in the same kidney. The cystic structures are by dilatation of the parts of the otherwise continuous nephrons, particularly the nephron loops, the loop of Henle. What happens is, as we have seen, the ureteric bud is going to penetrate the metanephrogenic blastema and results in the formation of the kidney tissue. But what happens, supposingly, at some areas, the ureteric bud, the divisions of the ureteric bud are not able to penetrate the they don't come in contact with the metanephrogenic blastema, and they are not only at some points they are in touch. For example, at 70% of the places they are in touch, so in that region proper kidney tissue will be formed, and in the rest of the 30% of the places the kidney tissue is formed, but it's not in contact with the the parts of the collecting system. So what will happen, since the kidney will start functioning, the urine production, the filtration will occur, and it will be keep on collecting in the uh, tubules of the kidneys, that is the proximal loop of Henle and distal convoluted tubules. They'll start dilating and start forming the cysts. So they will, and later on, as and when more dilatation occurs, those part of nephrons will stop functioning, and there will be a degeneration of those nephrons. And later on, only the 70% of the kidney will be functioning, and 30% will degenerate. And those here 30%, as we have already seen, the number of nephrons is fixed at birth. So only there is a compensatory hypertrophy of the remaining tissue, while the number of nephrons is going to remain the same. Ectopic ureter. An ectopic ureter does not enter the urinary bladder, which is the normal location of entry point of the ureter. In males, the ureter usually opens into the neck of the bladder or the prostatic part of the urethra. They may also enter into the less common locations, into the ductus deferens, prostatic utricle, or seminal gland. In females also, they can open into the neck of the bladder or the urethra or vagina or vestibule of the vagina. So in the diagram, we are going to see the ectopic locations of the ureter. And incontinence is the most common complaint resulting from an ectopic ureter, because the urine flowing from the orifice does not enter the bladder. Instead, it continuously dribbles from the urethra in males and urethra or vagina in females. So here we can see in the females, the more common locations of the ectopic urethral orifices is the trigone of the bladder, the bladder neck, the urethra, and the vestibule. The less common locations being the uterus, the cervix, and the vagina. Similarly, in the males, the more common locations of ectopic urethral orifice being trigone of the bladder neck, the prostatic urethra, and the less common locations being the vas deferens, the seminal vesicles, and the ejaculatory duct. Now, this is another anomaly of the ureter. Now coming to the development, we have seen the development of the kidney, which is from two in the beginning slide, which I shown, like the development of the kidney from the metanephrogenic blastema and from the ureteric bud, which we have already seen. Now coming to the development of the urinary bladder. The development of the for the development of the urinary bladder, we'll first discuss the cloaca. It is the expanded distal part of the hindgut. Hindgut, we have seen the kidney, which is a mesodermal structure, the nephrogenic cord, which is a mesodermal structure, whereas the gut system develops from the endodermal structure. So, cloaca, this is the distal part of the gut, distal part of the hindgut. It is closed below by a bilaminar cloacal membrane, that is the fusion of the endoderm and the ectoderm. Then later on, a mesodermal urorectal septum divides it into a primitive urogenital sinus ventrally and anorectal canal dorsally. So in this diagram, we can see this urorectal septum is developing, and it is dividing the cloaca. Initially, we have seen the common opening for the genital system and urinary system. Later, it is divided by urorectal septum, giving rise to the primitive urogenital sinus ventrally in the anterior part, and posteriorly or dorsally the anorectal canal. So this is now the urogenital sinus and the anorectal canal. So cloaca receives three openings: the allantois ventrally. Allantois is nothing but this one, which will undergo, the remnant of which will form the urachus and the median umbilical ligament. So cloaca receives this opening from apex, and the mesonephric ducts. These are mesonephric ducts, one opening on each side. As we have already seen, mesonephric ducts giving to ureteric bud and the formation of the kidneys taking place. So urogenital sinus or cloaca receiving all the three openings: urachus or the allantois ventrally, and the mesonephric ducts, one on each side. Now, the urogenital sinus, which is further divided into three parts, that is the vesical part that forms most of the urinary bladder and is continuous with the allantois in the apex region. A pelvic part that becomes the urethra in the neck of the bladder, the prostatic part of the urethra in males, and the entire urethra in females. A phallic part which forms the penile urethra in case of males. So the bladder forms from the vesical part of the, most of the bladder forms from the vesical part of the urogenital sinus, except the trigone. Trigone is formed, the area at the base of the bladder between the openings of the ureters is derived from the caudal end of the mesonephric ducts. I'll show this in diagram later on, how the trigone region is formed. And the entire epithelium, as we can see, the urogenital sinus is endodermal and the mesonephric ducts are mesodermal, and the trigone area is developing from the mesonephric ducts, whereas the most of the bladder is developing from urogenital sinus, which is endodermal. So most of the entire epithelium of the bladder is derived from the endoderm of the vesical part of the urogenital sinus. Even the epithelium at the trigone area, which is a mesodermal structure, it will also be later replaced by the endodermal lining. So entire epithelium of the bladder is derived from the endoderm. The other layers of its wall develop from the adjacent splanchnic mesenchyme. So the urinary bladder, now we see the development. The urinary bladder is developed from three sources: the vesical part, that is vesico-urethral canal, endoderm, major part of the urinary bladder and prostatic urethra, the proximal part of the allantois, which is forming the apex of the urinary bladder. Distal part gives urachus or obliterated median umbilical ligament. Absorbed proximal parts of the mesonephric ducts, which will form the trigone of the urinary bladder. So this is the trigone of the urinary bladder, which is formed by the absorbed parts of the mesonephric ducts. As we have seen, mesonephric ducts which open into the cloaca and gives rise to ureteric burden. So they will form at the distal end, they'll fuse and form this trigone region of the urinary bladder. So here we have shown the changing relationships of the ureter and mesonephric ducts. Initially, as we have seen, the ureters are formed by an outgrowth of the mesonephric duct. So here in this diagram, we can see ureters are formed by the outgrowth of the mesonephric duct, but with time, they assume a separate entrance into the urinary bladder. So they are going to assume a separate entrance into the urinary bladder, and the mesonephric ducts will descend down and will form in the fuse in the midline and form the lead to the formation of the trigone of the bladder. So the trigone of the bladder is formed by the incorporation of the mesonephric ducts. So this is the trigone area, which is formed. These blue color structures are the mesonephric ducts, which fuse in the midline and lead to the formation of the trigone of the bladder. Yeah. Now coming to the anomalies, urachal anomalies. It may be a fistula, it may be a urachal fistula, it may be a urachal cyst or urachal sinus. If it's patent all through, then on both the sides, then it leads to formation of a fistula. Very rarely, the entire urachus remains patent and forms a urachal fistula that allows urine to escape from its umbilical orifice. Or what happens is a urachal cyst can be formed. The remnants of the epithelial lining of the urachus may give rise to the urachal cyst, and there the fluid collection will take place and lead to the formation of a cyst. Urachal sinus, where one side of the entry is closed and other side is open. If the inferior end of the urachus may dilate to form a urachal sinus that opens into the bladder and the superior part is closed. In this diagram, we have seen that the lumen in the superior part of the urachus remain patent and it is open to form a urachal sinus that opens at the umbilicus. The development of the genital system. Now we have discussed the development of the urinary system. Now we are coming to the development of the genital system. Sex differentiation is a complex process that involves many genes, including some that are autosomal. But the key factor, the key gene in its the key to sexual dimorphism is the Y chromosome, which contains SRY, sex determining region on Y chromosome, on the short arm of Y chromosome. The protein product of this gene is a transcription factor that initiate a cascade of downstream genes that determine the fate of the rudimentary sexual organs. So in the beginning till 6 weeks, both they're not able to differentiate and there are indifferent gonads. And later on, the depending on the chromosome, the differentiation will take place into the male or female. The SRY protein is the testis determining factor, and under its influence, male development occurs. In its absence, female development is going to take place. Although the sex of the embryo is determined genetically at the time of fertilization, the the gonads do not acquire male or female morphological characteristics until the seventh week of development. Till then, there will be indifferent gonads. So, if there is absence of SRY gene, that is no testis determining factor, then development of ovaries and female duct system from the paramesonephric duct takes place. So, development of gonads is from three sources: the mesothelium, that is mesodermal epithelium, the underlying mesenchyme, and the primordial germ cells. Those are the undifferentiated sex cells. So here we can see in this diagram, this was our nephrogenic cord. This was pronephros, mesonephros, metanephros. This, at the anterior part of this mesonephros, there is a proliferation of this mesoderm epithelium and its underlying connective tissue forming the gonadal ridge. And here the gonadal ridge is going to form from where the development of gonads is going to take place, and the proliferation of this mesodermal epithelium and mesenchymal, which is later pierced by the primordial germ cells. Primordial germ cells through the umbilical vessel and through the hindgut and dorsal mesentery, they are going to traverse and reach the gonadal ridge, where they will take part in the formation of development of gonads. So this is the gonadal ridge and nephrogenic cord giving rise to kidney system, gonadal ridge forming the gonads, the ureteric bud, metanephrogenic blastema. I have explained this diagram in the previous slide also. So the initial stages of gonadal development occurs during the fifth week when a thickened area of mesothelium develops on the medial side of the mesonephros. So in the previous diagram, this is mesonephros, the middle part of the nephrogenic cord. This is nephrogenic cord, and the middle part of the mesonephros. There is this thickening, proliferation of tissue. The thickened area of mesothelium develops on the medial side of the mesonephros. Proliferation of this epithelium and the underlying mesenchyme produces a bulge on the medial side of mesonephros, the gonadal ridge. Finger-like epithelial cords, gonadal cords, soon grow into the underlying mesenchyme. So from the epithelium, the multiple gonadal cords will grow into the underlying mesenchyme. The indifferent gonad now consists of an external cortex and an inner medulla. In embryos with an XX sex chromosome complex, The cortex of the indifferent gonad differentiates into an ovary. The cortex takes part in the formation of the ovary and the medulla regresses. Whereas in embryos with an XY6 chromosome complex, the medulla differentiates into a testis and the cortex regresses. So the cortex is going to take part in the formation of the ovary, and the medulla is going to take part in the formation of the testis. In case of XX set of chromosome, the medulla is going to regress. In XY set of chromosome, the cortex is going to regress. The fate of the primordial germ cells. So these are large spherical cells visible in the fourth week among the endodermal cells of the umbilical vessels near the origin of the allantois. In the sixth week, the primordial germ cells arise from yolk sac endoderm and endoderm migrate to the gut and then through the dorsal mesentery to reach the gonadal ridge. Germ cells proliferate and uh, migrate into the cellular cords to become surrounded by supporting cells. Germ cells that fail to enter a cellular cord undergo degeneration. So here we have shown the migrational path of the primordial germ cells along the wall of the hindgut and the dorsal mesentery into the genital ridge. So this is the common system which was divided by urorectal system, the cloaca, the common system, allantois, and here from the posterior part of the hindgut and from the this is the dorsal mesentery. So through this path, the primordial germ cells are going to migrate, and as we know, this is genital ridge and surround near to it is mesonephros. So they'll reach here in the genital ridge and lead to the formation of the gonads. Similar thing is being shown in this diagram. Here, this is the notochord area. This is the cut section, transverse cut section, the first diagram. And here, somewhere here, the aorta is going to develop, and this is the notochord from the ectoderm. So here is aorta, the primordial germ cells migrating aorta, giving its branch to the glomerulus, glomerular capillaries, all those things. And from here, the renal system developing from the mesonephric duct, and here the gonadal ridge, which with its proliferating epithelium and the underlying mesenchyme, which will later be penetrated by the primitive sex cords forming the gonads. Here is the paramesonephric duct giving rise to later which we are going to discuss. So here is the diagram as we have already discussed. In case of females, the second diagram, B diagram, in case of females, the medullary cords will degenerate, cortical cords will develop, penetrated by primordial germ cells, leading to the formation of ovaries. And most of the mesonephros will degenerate. Only the few of the remnant structures from mesonephros will develop in case of females, epiphoron, paraepiphoron, and Gardner's cyst. And the paramesonephric duct will lead to the formation of the female genital organs. In the first diagram, the medullary cords are going to persist, whereas the cortical cords will degenerate, and the mesonephros or the mesonephric duct forming most of the genital organs of the male system. Similar thing is shown in this diagram also. The cortical cords will later be divided and form the primordial follicles. The primordial germ cells supported by the surrounding cells, granulosa cells, will form the primordial follicles in case of females. And these sex cords do not divide here in this case and lead to the formation of the male gonads. So coming to the development of testes. Testes determining factor induces the seminiferous cords to condense and extend into the medulla of the indifferent cord. In addition to testosterone, the fetal testes produce a glycoprotein, that is anti-Mullerian hormone or Mullerian inhibiting substance, which suppresses the development of the paramesonephric ducts, which form the uterus and uterine tubes. So AMH, which is going to suppress the development of paramesonephric duct, and lead to the formation of male system. The walls of the seminiferous tubules are composed of two types of cells. Sertoli cells, the supporting cells derived from the surface epithelium of the testis, and the spermatogonia or primordial sperm cells derived from the primordial germ cells. Yeah, this is a very important diagram. Here we can see the origin of the structures. The part shown in yellow developed from the urogenital sinus, the part shown in pink developed from the mesonephric duct, and the part shown in blue from the paramesonephric duct. As we all know, male system, most of the male system develops from the mesonephric duct, and the paramesonephric duct regresses in case of males. So here we can see from the urogenital sinus, most of the urinary bladder, that is the apex, this is urinary bladder, even the prostate gland from the which arising from the prostatic urethra, prostate gland also is developed from the urogenital sinus, the bulbourethral gland, and the urethra, prostatic urethra, and the penile urethra. So all these structures develop from the urogenital sinus, whereas the structure shown in pink develop from the mesonephric duct, including all the collecting system like here the rete testis, epididymis, vas deferens, then here the seminal gland, and ejaculatory, all of these develop from the mesonephric duct. The paramesonephric duct, which rudimentary remnant in case of males, gives rise to appendix of testis. That is a remnant of paramesonephric duct. Similar diagram will see in case of females also. And now coming to the development of ovaries, the cortical cords extend from the surface epithelium of the developing ovary into the underlying mesenchyme during the early fetal period. As the cortical
Can you
cord,
be a bit louder?
yes, ma'am. As the cortical cords increase in size, primordial germ cells are incorporated in them at approximately 16 weeks. These cords begin to break up into isolated cell clusters, that is, primordial follicles, each of which contains an oogonium derived from a primordial germ cell. So here's the diagram, and we have seen the medullary cords degenerating here, the mesonephric part of the, and here the cortical cords or the primordial follicles developing. So this is the atrophy of the mesonephric duct, the primordial follicle in the ovarian cortex containing primary oocyte, the aorta, the paramesonephric duct, which will later lead to the formation of the fallopian tube, uterus, and the cervix and vagina. These are degenerating medullary cords and the mesothelium of the ovary. Similar diagram, these cords dividing and then later lead to the formation of primordial follicles. The similar diagram is being shown in different cut sections. So we have already seen the part shown in yellow developing from the urogenital sinus, that developing shown in pink is developing from the mesonephric duct, and in blue the paramesonephric duct. In case of females, the most of the development occurs from the paramesonephric duct, and the mesonephric duct regresses and rudimentary and forms three structures that is shown in the diagram: epophoron, paraophoron, and the Gardner's cyst. Yeah, here the Gardner's cyst, paraophoron, epophoron. So the yellow part, the urogenital sinus, forming the same structures: urinary bladder, urethra, paraurethral glands, and the lower part of the vagina, forming this the lower part of the vagina. Urogenital sinus forming the sinovaginal bulbs and the lower part of the vagina. And here the paramesonephric duct, the structure shown in blue, forming the fallopian tube, uterus, cervix, and the upper part of the vagina, and the paramesonephric duct, which we have discussed. Now, the descent of ovaries and the fate of gubernaculum. The ovaries, like how the kidneys develop initially in the pelvic region, later on they migrate to the lumbar region, whereas the ovaries are first developing into the lumbar region and later on they are migrated to the pelvic cavity. The ovaries are also pulled down from the lumbar region to the pelvic cavity. They settle down slightly below the level of pelvic brim, and the gubernaculum helps in the descent of the ovaries, and later on it forms two rudimentary it helps in the formation of two ligaments. That is, in this diagram we can see the ovarian ligament and the round ligament of the uterus. These are the two remaining structures which are derived from the gubernaculum. And the broad ligament is basically a derivative from the peritoneum. The development of female genital ducts, the mesonephric duct of the female embryo regresses because of the absence of testosterone. The paramesonephric duct developed because of the absence of Müllerian inhibiting substance. Female sexual development during the fetal period does not depend on the presence of ovaries or hormones. Later, estrogen produced by the maternal ovaries and the placenta stimulate the development of uterine tube, uterus, and the superior part of the vagina. The paramesonephric duct forms most of the female genital tract. The uterine tubes develop from the unfused cranial part of these ducts, and the caudal part fuse, forming the fused portions from the uterovaginal primordium, which gives rise to the uterus and the superior part of the vagina. The endometrial stroma and the myometrium are derived from the splanchnic mesenchyme. The same diagram is being shown here. The cortical cords, degenerating medulla, mesonephric, paramesonephric duct, mesonephric duct regressing, remaining structures from mesonephric duct, epiphoron, paraepiphoron, and catenus. And the uterus, the paramesonephric duct, the pink one, giving rise to the unfused cranial portions, giving rise to the uterine tube, and the fused caudal portions, giving rise to the uterus, cervix, and the upper part of the vagina. Now coming to the congenital malformations of the uterus. So in case the both of the paramesonephric ducts fail to fuse, then in that case we have the condition uterus diadelphys with double vagina. And if the paramesonephric ducts fuse in the lower in the fail to fuse in the upper part and they fuse in the lower part, then it gives rise to the condition uterus bicornis. And if they fail to fuse longitudinally more higher level, then only a arcuate uterus arcuate uterus is left. And uterus bicornis, when a rudimentary when one of the paramesonephric ducts fail to develop properly, then it gives rise to uterus bicornis unicollis. Development of vagina. The fibromuscular wall of the vagina develops from the surrounding mesenchyme, contact of the uterovaginal primordium with the urogenital sinus. Uterovaginal primordium derived from the paramesonephric duct, and the part from the urogenital sinus forming the sinus tubercle induces the formation of paired endodermal outgrowths, that is the sinovaginal bulbs. They extend from the urogenital sinus to the caudal end of the uterovaginal primordium. The sinovaginal bulbs fuse to form a vaginal plate. Later, the central cells of this plate break down, forming the lumen of the vagina. We'll see in the diagram here the lining of the urogenital sinus. So this lining of the urogenital sinus will proliferate and will reach the lower part of the uterovaginal canal, forming the sinovaginal bulb. And these, along with the lower part of the uterovaginal canal and the upper part, the lower part from the sinovaginal bulbs, will form this vagina, which will later canalize and form the lumen of the vagina. So uterovaginal canal, that is a mesodermal structure, whereas urogenital sinus endodermal. The upper part from the mesoderm and the lower part from the lower part from the urogenital sinus, that is the endoderm. Uterovaginal canal, mesoderm, in contact with the lining of the urogenital sinus, sinovaginal bulbs are formed from proliferation of endodermal lining. Solid vaginal plate derived partly from mesoderm of uterovaginal canal and partly from endoderm of sinovaginal bulbs. Vagina formed by canalization of vaginal plate. The fornices and the upper portion of the vagina are formed by vacuolization of the paramesonephric tissue, the paramesonephric tissue, and the lower portion of the vagina is formed by vacuolization of the sinovaginal bulbs. Same thing is being shown in this diagram. Now coming to the anomalies of the vagina, vaginal atresia. When there is a failure of canalization of the vaginal plate, it will lead to the formation of, it will lead to condition vaginal atresia. And imperforated hymen, if the middle portion of the hymen fails to get thinned down during the last weeks of intrauterine life, there would be a failure of rupture of hymen during the time of birth, resulting in imperforate hymen. Now, coming to the last slide, that is the development of the external genitalia. External genitalia. So this it is stimulated by estrogens. The genital tubercle elongates only slightly, forming the clitoris. Here we can see this is genital tubercle forming the clitoris. The urethral folds do not fuse in case of females and develop into labia minora. So these are the urethral folds forming the labia minora. The genital swellings of the labioscrotal swellings, these one enlarge to form the labia majora and the urogenital groove, which is open and forming the vestibule. So that's all for today's seminar from my side. Thank you.
Thank you very much, Nisha. That was very, very excellent presentation, I must say. Thank you so much.
Thank you, ma'am.
And I think we all enjoyed your presentation. PGs, do you have any questions? Any questions? Hello? Can you hear me, Nisha?
Yes, ma'am, I can hear you.
Okay. So any questions from your friends? Hello?
Yes, ma'am.
Okay. So I think, Nisha, you have done a very good job, told us about the development of the urinary tract, the genital tract, the migration of the eggs from the yolk sac down the dorsal mesentery into the genital ridge, and how the male and the female, the mesonephric and the paramesonephric ducts, etc. So I think we are all very well understood with this. And with that, I thank you, Dr. Nisha. And all of you should stay on. And I request all consultants also to switch on for Dr. Dinesh Kaul's lecture. Thank you, Nisha. Thank you, Dr. Mamta.
Thank you, ma'am.
Thank you. Thank you.
Mamta, can you tell Nikki to tell all the consultants to join in if they haven't joined in? Whoever has not joined, because there's going to be a very interesting lecture by Dr. Dinesh, and I think all of us have a lot of questions to ask.
I will tell her. I will tell Shalu to inform everyone once again.
So maybe we can join back after 1 minute. Dr. Dinesh, are you
Yeah,
there?
sure. It's fine. If you want to take a break for 5 minutes, in the meantime, maybe you can send a mail to academics. They can circulate it.
Sure.
Practice about the timelines, the COVID vaccines, what are in the offing, and what we would expect in the near future. That was way back in 13th of July, when none of the vaccines except the Chinese vaccine was launched. So this is an obituary to him. I think my okay, what will be talking about the COVID-19 vaccines. I'll walk you through a little bit about the immunology of the vaccines, how the vaccines work. We'll talk about the vaccine candidates, what were there in the preclinical stage, the potential vaccines, what are at present in the clinical trials or which are already launched, which have got the FDA and the US government approval or the Indian government approval. And then finally, we definitely need to talk about the national policy for COVID-19 vaccines in India. And because this is really of concern, because there is from 16th of January, the nation is going to launch the biggest vaccination program done anywhere in the world. So we'll be talking about. Let's have a little about the epidemiology. Most of you must be checking who are really interested about the facts and figures, how many cases we had. If you see the comparison between the two these columns on 17th of September and 8th of January, we had definitely the active number of cases had increased, had decreased. The number of people who were discharged home from 4 million to about 10 million had been discharged on 8th of and but the total number of cases have obviously increased from 5 million to 10 million. And obviously we would expect the deaths had increased in these 3 or 4 months. They must have almost doubled. So about 150,000 people have already died in India because of the COVID disease per se. But definitely you can see the figures are the death figures in last few weeks have gone down. If we see the whole world, there are about 85 million people still who were who were affected with COVID, and India accounts for about 12% of the total number of cases. But the number of deaths is highest in USA. About 18% of the total deaths are in USA. Fortunately, we have less number of deaths as compared to rest of the countries. Still we account for around 8% of the total COVID deaths in India, of all over the world. So let's move to the next slide. We have a lot of these viruses have clades. So over the time, even with the new virus, this happens with the influenza virus, this happens with other viruses, they mutate. So some of these mutations are pathogenic, some are not pathogenic. So if we see that there is this mutation, D614 mutation. So this is the original Wuhan virus had more or less mutated to this clade, and this is the clade which is circulating in India, in rest of the world. And then recently we had a lot of concern about mutations in the UK, what we call as the variant of unknown variant mutation, the VOC 2001 mutation. We'll be talking about this, but this is a phylogenetic tree. You can see from the original genomic sequence, there could be a lot of variability in the viral sequences, but some can be pathogenic, some are not pathogenic. In India, we have these sort of clades which are circulating. So these are clade A2A, 0, A3, B4, which have been isolated from the Gujarat Institute of Cellular Molecular Biology. And what is in the southern states is the clade A31A, 31I. So this is the circulating strains into the southern states, which has been isolated by the cellular and molecular biology. Fortunately, we have very wonderful molecular biologists who are working all the time and isolating these strains. That has in fact led to development of a new vaccine in India. So let's see these mutations. So this is the virus with D614G mutation. So this is the mutation which is circulating in most of the parts in the world. Most of the viruses are having this mutation. So there is an NS1 non-structural 1 protein encoding mutation, but it is not really that pathogenic. So prior to development of we had some mink coronaviruses occurring in Denmark. So minks are basically mammals, and their fur is reared for the making mink blankets and all that. So unfortunately, this mink coronavirus did not go to the humans, and this remained, but it was definitely of concern way back in January and in last December that it might go to the humans. What is of concern is the UK mutation at this point. So this is the UK mutation, which is in the news, VOC 20201 variant. So this is supposed to be more transmissible. So that's why patients who are, or rather people, passengers who are coming from UK, irrespective of their COVID PCRs, positive or negative, they are being given home, hospital and institutional quarantine. They are not let go home. So that's the concern. But we don't know that whether this UK variant is more pathogenic. There is no data to suggest that this is more pathogenic. Yes, it is more transmissible. And then of recent concern are certain mutations occurring in South Africa. That is the 501V2 mutation variant. So that's one more that is present in South Africa. So WHO has notified about 5 clades of these SARS coronaviruses, and about 11 mutations have been reported worldwide as on August 22nd. And later we have the other mutations. Now let's come to the immunology of the vaccines, for how really the vaccines work. This is a schematic diagram I have taken for a, this is basically a polysaccharide antigen. Say if you are injecting a vaccine, a DPT vaccine or a typhoid vaccine, polysaccharide antigen, it goes into the body, you inject it intramuscularly or subcutaneously or by any other route. The B cells will recognize it, this antigen, and they will produce antibodies, the IgG antibodies to this polysaccharide antigen. So over the time, this polysaccharide antigen will evoke an antibody response, which will decline over time within few weeks to years, depending upon the antigen. And then this will die down over some time. So if we have to make this more immunogenic, so what the vaccine biologists do, they make this polysaccharide or antigen, protein antigen, more immunogenic. They will conjugate it with an tetanus protein, with a diphtheria protein, or with some other bigger molecule like aluminum hydroxide, so that it is bulky. With the bulk, with increasing the molecular weight, it is more immunogenic. So it evokes both a B cell response and a T cell response. There is a complicated signaling pathway between the B cells and the T cells, where they communicate with each other with the different interleukins, and they produce different antibodies. So you get both immunoglobulins, plus the T cells have also been incorporated, so that there is a predominance of a CD4 positive T cells, and they are recruited in generating a very good immunogenic response. So this is how, if we give a conjugate vaccine, this is the immune response. But if you boost it after say 8 weeks or 12 weeks with the polysaccharide antigen, this is true for a typhoid vaccine, it will evoke a very good immunogenic response. Now, you must have heard about the newer these messenger RNA vaccines. So conventionally, we have been taught about the live attenuated vaccines. We have been taught about the kill vaccines. We have been taught about the subunit vaccines. But then lately, for the last 20 years or so, we have had DNA recombinant vaccines or RNA recombinant vaccines. But lately, we have heard this term, the messenger RNA vaccines. So there are a lot of viral vector vaccines. Viral adenoviruses are used as vectors to produce vaccines. And same way, there's a newer technology called a messenger RNA vaccines. So what is this messenger RNA vaccine? So you have the genetic code, while being translated from the genes onto making the proteins, the messenger RNA has the code to making different proteins. So if you take this messenger RNA, which is designated for a proper vaccine protein, so if it has a particular genetic code, it is enveloped in certain nanoparticles, okay, lipid nanoparticles. So it goes into the body once it is injected, and the B cells will take it. This messenger RNA directs the formation of immunoglobulins, which is directed against a particular protein. Here, for the coronaviruses, it is directed against the the spike protein, the S protein, or other specific antigens. It goes into the cell, but it doesn't enter the nucleus. The common myth is that it will enter the nucleus, it will incorporate into the human genome and have some disastrous effects. But that's not so. It is in the cytosol and it gets destroyed by the cellular machinery. But at the same time, it produces very good immunoglobulins. This is the basis of this newer messenger RNA vaccines. Okay, then you have certain other antigen presenting cells. Basically, for the B cells, the T cells will act as antigen presenting cells. So they can carry the antigenic epitope onto their surfaces. They present it onto the B cells to produce the immunoglobulins. The whole idea of the vaccine is to produce protective immunoglobulins and have very good T cell response. So that's what is the main function of the vaccines. Now, there are at least 56 vaccine trials which are being done all over the world. Some are completed, some are still in the phase 3 trials. But then in addition to that, there are about 166 vaccines which are still in the preclinical stage on the experimental animal models. So we hope we should be getting few more vaccines in the few years if the epidemic continues, or otherwise the research will die down on them. So you have the viral vector vaccines. They can be replicating or they can be non-replicating vaccines. And then you have the viral vector vaccines with antigen presenting cells. You have the protein subunit vaccines. You have the nucleic acid vaccines. You have the DNA recombinant vaccines. As you know, the hepatitis B is a DNA recombinant vaccine. Or you have these Moderna's messenger RNA newer vaccine, which I discussed. And you have the live attenuated vaccines. Say like the oral polio vaccine is primarily a live attenuated vaccine. The measles vaccines, the rubella vaccines, the mumps vaccine is a live attenuated vaccine. Okay, or a BCG is a live attenuated vaccine. Then there are old age technology is the inactivated vaccines. So DPT, that's how they have used, the Bharat Biotech has used the technology of using conventional DPT manufacturing technology to make this inactivated vaccines for the COVID. So they can be given in various dose schedules. So this is a little repetition of this same. I have taken this from the Dr. Chug's presentation from the CMRP. You can go through this CMRP article. It is a very good article for at least for the postgraduates to understand the basic biology of the vaccines. So lately we have, I may go back, this is what we are using. BCG, we thought we could use it as a repurposed vaccine. This was meant to prevent TB, but there were a lot of clinical trials being done to see that whether we can really use BCG to prevent the at least the mortality and the morbidity of the disease. So there are certain studies which have shown a little beneficial role of BCG given in the elderly population. So it will prevent from getting severe sepsis in such individuals. Now let's come to what is of interest is the COVID-19 vaccines. So certain vaccines have been given regulatory approval, but certain vaccines are pending for the approval. They have put their application for the various drug controller agencies. So the first vaccine, at least which was developed, was this messenger RNA vaccine. This is called the mRNA. They code named it initially mRNA 1273 vaccine. The Moderna is a company, which US-based company, which collaborated with the NIH, and now this has got an FDA approval. They have shown wonderful phase 3 trials. Second vaccine, messenger RNA vaccine, is the mRNA vaccine from the Janssen's. This is an adenovirus vectored vaccine. Okay, I will tell you a little bit about the adenovirus vector vaccines. Then third vaccine is the messenger RNA vaccine, is of the Pfizer, which is very much in the news, which was given approval on 1st or 2nd of December by the US FDA. Subsequently, the UK health authorities also gave the approval to this vaccine. But unfortunately, there are very few trials of this in the Indian setup. And then this is the first vaccine, the adenovirus vector vaccine. This is different from the messenger RNA vaccine. It uses adenovirus as a vector for the development of the vaccines. This is what the CanSino Biologicals and the Beijing Institute of Technology use this vaccine technology in China. And they used it before really they could go to the phase 1 trials. Now they are doing phase 2 and phase 3 trials are being done in Russia. But in fact, this vaccine changed the course of the disease in China, as you know, that they could really contain the epidemic. Now coming to further with the COVID-19 vaccines, this is the adenovirus vectored vaccine, the Oxford AstraZeneca's vaccine, code named as AZD1220. Okay, but this will be named as Covishield because the AstraZeneca people are collaborating with the Serum Institute of India to develop this vaccine to manufacture this vaccine in India. And this is the vaccine which the government is going to purchase and going to the healthcare individuals over next few months. This is one more vaccine. You must have heard that the Russia launched the vaccine way back in August, and in fact, the Premier Putin's daughter was the first to get the vaccine, and Premier Putin also got the vaccine. So this was the military institute in Russia, developed in collaboration with the scientific world in Russia. This is a dual adenoviral vector. So there are two adenovirus vaccines which are used in this vaccine technology. So this was named as Sputnik V, and the Gamaleya Institute of Technology is the research institute which developed this vaccine. And then there is our indigenous vaccine made in India, the inactivated SARS-CoV-2 vaccine with the brand name of Covaxin. The Bharat Biotech has launched this vaccine, developed this vaccine. ICMR, in fact, developed this vaccine. The National Institute of Virology, Pune, developed isolated the strains of the coronavirus initially somewhere in way back in March and April. Then with the collaboration of Bharat Biotech, they have developed this inactivated or the killed vaccine. This is also going to be used in the Indian setup for the vaccination of healthcare individuals. Now, this is a slide which has been made by the government agencies and the WHO with assistance from WHO. This is more or less a teaching slide which they are showing to different trainers in different programs. These are some of the vaccines which India may have in the near future. Bharat Biotech vaccine is already approved. the Cadila vaccine is in the phase 2 trials. The phase 3 trials are still underway. Phase 2 have been completed. The University of Oxford and the AstraZeneca vaccine, the Covishield, what we call in India, is already approved by the Indian drug regulators. Also, the US FDA approved, and also the UK health authorities approved. This is going to be a good vaccine. And then this is the vaccine Sputnik V. In fact, certain pharmaceutical companies are going to collaborate with the Gamaleya Institute. They are going to have the transfer of technology to develop this vaccine in India, so that we should be having this vaccine also in India. Now let's come to various vaccine trials which are being conducted in India as of now, or which have been finished. In fact, Gangaram Hospital, under guidance of Dr. Anupam Sachdev, collaborated with the Bharat Biotech to do the phase 3 trials of Covaxin at Gangaram Hospital, though he was involved a little late. So they have, I will show you the data of each of these vaccines which are important in the Indian setup. So this is the Covaxin inactivated vaccines. The Covishield is basically an adenovirus vectored vaccine. It has been developed in the chimpanzee cells using an adenovirus vaccine. This is the code name of the vaccine before it was launched. The brand name is Covishield. There is one more company, the Zydus Cadila, is working on a plasma DNA vaccine. So phase 3 trials are still underway. I have talked about the Sputnik vaccine, the Russian vaccine, and then the Biologicals E is also going to develop a vaccine. They are also under phase 3 trials. This should be also, because Biologicals E is an Indian company, so we should be seeing this vaccine in the Indian market. Now, before we go to different vaccines, to each of these assets and demerits of these vaccines, what is important for as an immunologist is that safety is the top priority for the vaccine. So we don't want people to get sick with vaccines. This happens way back in the polio vaccine, inactivated polio vaccines in 1940s, when there were a lot of paralytic poliomyelitis with the vaccine, obviously because of the contamination. So we have to see that our protocols are in place. We have to report the adverse effects, and serious adverse effects is mandatory. Normally, in a clinical trial, we report only serious adverse effects, okay, within 24 hours. But here we are supposed to, just give me a minute. We are supposed to give details about all the adverse effects of these experimental vaccines. Now, who needs to be vaccinated first? So healthcare personnel who have the risk of direct or indirect exposure to the patient or infectious material need to be get vaccinated. All long-term care residents who are in nursing homes, especially in US, need to be vaccinated. So the elderly population is vulnerable. But the Indian guidelines are a little different. These are the Indian guidelines. The NAGVAC is the authority which gives recommendations for these vaccinations in India, especially the COVID vaccine. They have said that healthcare individuals plus persons who are above 50 years. Dr. Jyotha, just give me a minute. Yes, sorry. So persons above 50 years and all persons who are below 50 years of age need to be with comorbidities. If they have diabetes, if they have hypertension, they need to be vaccinated. And obviously, all healthcare individuals will be vaccinated. Am I audible? Okay, I am not mute. Fine. So let's come now to each of these vaccines so that I will tell you about the merits and demerits of all these vaccines. The first coronavirus, which was approved in China, was the adenovirus vectored coronavirus vaccine. Manufactured by the CanSino Biologicals is the research company which made this vaccine with collaboration with the Beijing Institute of Technology. And though they launched this vaccine immediately at the height of the epidemic, but now the phase 2 and 3 trials are underway. Phase 2 trial has been completed already, okay, and in China, in Wuhan, and they have shown very good results. I'll show you the results. Phase 3 trials are presently being done. I think they have been completed in Russia with about 40,000 participants. So that's a quite sizable number for a vaccine. So let's see, this is a little summary of the trial of this Sinovac, the Chinese vaccine, adenovirus vector vaccine. So they use different doses of this vaccine. So they found out that if they were using a higher dose of the vaccine, it was more immunogenic. It would have a very good antibody response. The seroconversion was about 96% with these vaccines, and they got a very good T cell response. So how they checked the T cell response? They did a test called a gamma interferon assay. The way we do quantiferon gold assay, same way they tested the T cell integrity by doing a gamma interferon assay, and they found out that it generates a very good T cell response. Okay. So the serious side effects were greater in the population who received a higher dose. So that's expected. About 9% had serious adverse events, while as 1% has serious adverse in the lower dose. But if you see, the higher dose is more immunogenic. So you have to have a balance. So this was a story about the Chinese vaccine. Now coming to our vaccines which are of interest in India. So this is the Oxford Zeneca's vaccine, which is being launched in India. The Serum Institute of India is going to manufacture this vaccine. In fact, they have manufactured millions of doses of this, and they have been shipped already to different vaccine storage areas all over the country. They will be done within few days. So this is a chimpanzee adenovirus vectored vaccine. Okay, this is a genetically engineered vaccine where the genes are coding the adenovirus has been taken as a vector. So you splice the genome of the adenovirus. You put the genome of which codes for the spike protein or the NS1 protein. So it will subsequently, when this adenovirus will multiply, it will produce copies of this protein. So this is the basis of the vectored vaccines. So whatever cumulative data we have till now from the 4 randomized controlled trials is quite promising. I will show you there is an interim data which has been generated recently, and it is which has been published in 9th January issue of Lancet. It has shown very promising results. The efficacy data is about 70% is the cumulative efficacy data of this vaccine. So this is given in a 2-dose schedule, 0 and 28 days. Here they used a different strengths of the vaccine, and they compared it with the placebo or a meningococcal conjugate vaccine. They found out that yes, in about 11,000 odd participants, it had a very good immunogenic response. 87.8% had very good immune response with the Oxford vaccine. So the vaccine efficacy, as I said, after 14 doses, after the second dose was about 70.4%, the cumulative vaccine efficacy. There was a little problem with their vaccine. They gave half the dose to some of the population initially, and then they doubled the dose. They found out that even If you give initially the half the dose, subsequently you give a full dose, you have a very good immune response. Okay, and so if you see that this in the control in the test group, those who got the vaccine, 30 just 0.5% of the individuals got infected in spite of getting the vaccine. Vis-a-vis in the control group, about 1.7% of the population got infected who got the vaccine. So this is quite statistically significant. So that's how this comes to a vaccine efficacy of 70.4%, and that's how it has got. And obviously, they have analyzed other data, the safety data, the efficacy data, the seroconversion data, and it has been given approval by the US FDA, the UK health authorities, and the Indian drug regulatory people. So that, as you would expect, that you do get adverse events with a lot of vaccines. So there were about 84 serious adverse events with the vaccine group, and whereas there were 91 in the placebo group. So this is more or less similar. You had one case of transverse myelitis after 14 days after the second dose in the vaccine group, and then they had about 2 cases of transverse myelitis in the vaccine group, and there were about day 10th, and then in the control group they had 1 case of transverse myelitis. But this is one of the serious side effects of this vaccine, but this is quite low. Okay, so this is getting a Guillain-Barré syndrome or a transverse myelitis occurs with influenza vaccines also. Say like 1 in 100,000 is the incidence of getting GBS with an influenza vaccine. So here this is a controlled data from a small group of population. So we can't say that how many people will get transverse myelitis or GBS. It is only when we use this vaccine in the public, only we'll get an idea. The cost is going to be less, just $2 to $3 for this vaccine. In India, most of the healthcare workers are going to get it free of cost. Now coming to the recombinant adenovirus, which is using a dual adenoviruses, 26 and adenovirus 5. This is the basis of the Russian vaccine, the Sputnik V vaccine. So instead of 1 adenovirus vector, they are using 2 adenovirus vectors, and phase 1 and 2 trials have been already completed, and they have also shown a very good safety profile and a good humoral and a cellular immune response. The advantage of this vaccine is that it is a freeze-dried formulation, unlike the Pfizer vaccine, so you don't need a strict cold chain. So possibly that's why the government is very keen to get this vaccine also in the Indian scenario. So this is a little, I will not go into data of this. Those who are interested in looking at the scientific details about the phase 1, phase 2 studies, they can log on to the safety and immunogenicity of the Sputnik. This has been published in Lancet of September 26th issue. Now coming to the Moderna vaccine. This is a promising vaccine which was developed very early in the epidemic, way back in March and April in USA. So one of the companies, the Moderna TX, collaborated with the National Institute of Allergy and Infectious Diseases. This is a NIH institute in US where they formed a unique vaccine. They had known about this technology of the messenger RNA nanolipid technology, but it had not been used in earlier vaccination strategies. So this is a newer technology for us, but whatever data what we have for this in phase 3 trials is quite promising, and we should be using this vaccine, though this is not available in India as of now. The efficacy is very good. This is as efficacious as the Pfizer vaccine. About 94% is the efficacy of the Moderna vaccines. The adverse reactions will be relatively low as compared to the other vaccines. Just a pain at the local site, little headache people had in the experimental group, but there were no serious life-threatening or allergic reactions. They did not have any cases of transverse myelitis or GBS. Okay, but we are little people, lay public are little skeptical about the nano particle technology because we have not used it in the bulk manufacturing of the vaccines as of now. So we need to see this how it works. But and this can be stored at minus 20 degrees in the freezer for 6 months and at 4 degrees at 30 days. So we should be seeing this vaccine in the Indian scenario also. Now coming to this is the I think, yeah, this is the Pfizer vaccine. These were the two different vaccine models which the Pfizer used. The BNT 1621B and BNT 162B, and then they subsequently found that they would be using this vaccine candidate in phase 3 trials, though they found out that with a higher dose, the fever and the immunogenic reactions were more, but this is the vaccine which is being used in the Pfizer vaccine. So just a little word about this is about the safety and immunogenicity data about different formulations of the Pfizer vaccine. They have just sort of clicked on the 100 microgram dose which they will be using in their final phase 3 trials, and this has been shown to be quite immunogenic. Now coming to the Moderna vaccine. The Moderna vaccine has shown a good response with this strategy. This is a little correspondence which was seen put in the New England Journal of Medicine. I'll not go into detail of this. This is again regarding the Pfizer trial where they have shown that the efficacy is about 95%. But the problem with the Pfizer vaccine is that you need to store it at minus 70 degrees. So we have a problem of logistics in India of the cold storage and the delivery problems, and obviously the cost. This is costing around maybe more than 2,000 rupees in the Indian market if it will be launched. But this is, as of now, the government is not going to buy this vaccine for in the Indian population. The adverse reactions I have mentioned about that. Lot of adverse reactions were seen in the lay media where they found out that there were a lot of nurses who got but this is true of other vaccines. So we will not, you can get hypersensitivity reactions to any of the vaccines. So you should be ready with the anaphylaxis kit. You should have hydrocortisone and adrenaline ready if you are going to vaccinate in a public health campaign. Now coming to the old indigenous made in India vaccine, the Bharat Biotech vaccine, the whole virion inactivated SARS-CoV-2 vaccine. This is, as I said earlier, this was the inactivated vaccine derived from the SARS-CoV-2 isolates, which were initially taken from National Institute of Virology. The technical know-how was taken by the Bharat Biotech because they have the facility for manufacturing vaccines. Phase 1 and 2 trials have been completed. Phase 3 trials have already been completed with 22,000 odd individuals all over India in different centers in India. Okay, instead of 28,000, just about, because they felt this was at a particular juncture that they were going to get the approval. So they have got incorporated about 22,000 odd individuals. So this was a double-blind, multicentric trial. But here, as of now, they have evaluated just the reactogenicity and the immunogenicity. We don't have the data about the efficacy of the Bharat Biotech vaccine as of now. But the one plus point about this vaccine is that it is an old age technology, which the kill virus manufacturing technology, which the Bharat Biotech is using, or Serum Institute of India or other vaccine companies are very adept using these. So this is a novel, they are using a novel coronavirus. They are using a particular aluminum L-hydroxyquim. Conventionally, we are using aluminum hydroxide as an adjuvant in the vaccines, in the DPT vaccines. Here they have changed the adjuvant. This is the adjuvant. They have got this technology from a Kansas-based company, and they have shown that it is evoking a very good Th1 response. So possibly there is a theoretical proposition that this will work against very mutant strains also, because this is not directed against a particular spike protein or a NS1 antigen or any other specific epitope of the virus. This is a data which has been published, though this is not a peer-reviewed data. There are preprint servers. The MedRevix is one of the preprint server where they have shown that the data has been published of the phase 1 and phase 2 clinical trials. And this is a little bit about the data where they have shown that they have used various dose strengths. They have found that 6 microgram dose of this vaccine is quite good, though it is more immunogenic, it is more reactogenic, it has more adverse reactions as compared to the low dose. But the 6 microgram dose evokes a better neutralizing antibody response. So that's why the 6 microgram dose is being used in the Covaxin formulation. Fortunately, it has already got the drug controller's approval, though we don't have the data with the phase 3 trials as of now, but whatever data has gone to the drug controller with the animal study phase 1 and 2 data, the approval has been already been given. So let's hope this is a promising vaccine in the Indian setting. This I have already mentioned about how many people were incorporated. This is still underway. They are still trying to analyze the efficacy data. So maybe in near future, we should be having an efficacy data about this vaccine. Now coming to the public health strategies. So as you will be interested in that, either the Covaxin or the Covishield, the Bharat Biotech's Covaxin or the AstraZeneca's collaboration with Serum Institute of India, the Covishield, will be launched in the Indian setup, and with 16th of January, it is going to be rolled out in different health centers. I will go through, this is important, how this is being launched. The Government of India has formed a national program for COVID-19 vaccination strategy. The Ministry of Family Welfare with other collaborations, WHO, UNICEF, Bill and Melinda Gates Foundation has collaborated to formulate this program, and a training program has been done for different healthcare individuals, so that about 3 million, I think this should be more than that, about 30 million people will be given the vaccine in phase 1. So there is a registration system. It is not that everybody will get the vaccine. First, the healthcare workers will get the vaccine, and those who are in the front line, the police, the CRPF personnel, who are at the front line of COVID will get the vaccine. So their names have already been given by various authorities. They are registered in a system called CoWIN. This is the platform, computer vaccine intelligence network for the coronavirus, computer-based system. So the data is there, and the tracking of the viruses to the delivery of the viruses will be done through this COVID system, and only pre-registered beneficiaries will be vaccinated. As I said, that the recommendations for the vaccine have been given by the National Expert Group of Vaccine Administration, that is the NAGVEC, what we call it for short term. Dr. Paul, VK Paul has given the recommendations for the NAGVEC. This is a training slide where we are supposed to tell you about the setting up and conducting a training session, how it will be done in the community. This is very important, that we have to identify the training sites, say like in Gangaram hospital, in different government and private hospitals, in all government hospitals, primary care health centers, they will be taken as vaccination session sites. And the vaccine logistics and planning has been already done by the government. They have linked the vaccinators, the vaccinators and the beneficiaries will be linked to the COVID platform, and the vaccination session site structure and the flow chart of the beneficiaries is in progress. So this is how a vaccination site will look like. This is a schematic, diagrammatic expression. So say this is a site at a Gangaram hospital. You will be the person who is already registered on the COVID platform. So he will go in the vaccinating room where his credentials will be checked by a security person. This is designated as vaccination officer 1. Then his Aadhaar card or his details will be checked. He will be verified whether he is the same person. Then he'll go in the vaccination room. Here his temperature, everything will be checked with the whatever COVID precautions are to be taken. And then he will go in the vaccination room. Again, the vaccination officer 2nd, a nurse or a medic will cross-check whether his name is there in the COVID platform. He will show his valid ID proof to the vaccination officer. Then he will get vaccinated. Okay, one at a time. Once he will be vaccinated, he will go into the observation room where he will be observed for 30 minutes for any side effects. Okay. And at the same time, it will have the facility for the drinking water and the toilet. This is healthcare regulatory for the observation areas. Then this will be done, supervised by a vaccination officer 4, so that they don't see any crowding, clustering of people, and the credentials are checked. And finally, this will be all overlooked by a medical officer. So the whole system of vaccination will be entered in a computer software or a mobile phone, where every data entry of the vaccine given is made, and the person, the healthcare, will get an SMS on the mobile phone that he has been vaccinated on this date, and the next due date will be on such and such day after 28 days. So this is again the same diagrammatic expression showing how a COVID platform works. So this is a mobile handheld. So this COVID mobile application is not, you can't log into this. It's only the government officials who have the access to this COVID platform at this point. So they will log on to this platform, check the data entry will be made, the temperature entry, everything will be done electronically, and it can be monitored from one point. It will go into a computer network server, the cloud server, which will be connected through a web interface. So anybody who is authorized can log into the system and see what is happening across different centers. So this is something in a nutshell about the the COVID vaccination strategy. We will be also looking at the adverse reactions, because in the general public, in fact, even in the healthcare people, non-medicos, is a lot of adverse. There's a fear that this vaccination strategy might be causing a lot of adverse reactions. So they will be monitored for adverse reactions, and their data will be taken and generated onto the system for any adverse reactions. But we'll be, as medical people, will have a crash cart available in the vaccination centers. Anybody having any type of an immediate anaphylactic reaction will be taken care of, or will be, if they have a delayed type of a reaction, they can talk to their vaccinating officers. That data will be captured in the COVID platform, and a robust generation of data will be available for future use. So we need to talk about the AEFI. So this is a term which the public health people are using, the adverse event following immunization. So we are all for the vaccinators. We know this term AEFI, but so any untoward medical occurrence which follows after vaccination, it may or may not have a causal relationship with the vaccine. So the vaccine may be actually responsible for the adverse events, or it may not be. It is taken as AEFI. So anybody, if any X, Y, and Z gets the vaccine, next day he has a road traffic accident, that is taken as a AEFI. If he has a fracture, that is taken as a AEFI. If he has a fever, that is taken as a AEFI. So it is later for us to decide what is favorable and unfavorable, what it is really linked to the vaccine or not. Sorry. I'm sorry, I'm just a little, I'm sorry, I'm just a little out of Zoom app. Let me see if I can. Anyway, this is the, I'm trying to, I have lost the screen. Just a minute.
Sir, you can stop sharing and share once again.
Yeah. I think that I'm done with my last slide. So this is all what I wanted to about the public health. Okay, I'm back. Fine. Sorry. So this is the reporting system of the AAFI. We will be reporting the deaths, the inpatient hospitalization, any disability, any clustering of events in this COVID platform. That everything will be recorded. So even the minor ailments, the severe reactions will be recorded in the system. So we are supposed to I think the figures are wrong. So there will be more than initially. This was the strategy to get vaccinated about 1 million healthcare workers, about 2 million frontline workers, and later about 27 million people. So this is a colossal exercise which will be done. So this is a New Year's gift, possibly, which the health authorities have given to the people of India. So let's hope we utilize them, and we do have little decrease in the epidemic. But at the same time, we will not have to forego the use of masks and the preventive precautions. So all the vaccines have been, as such, they are in the sort of an EU emergency under authorization mode. This is called accelerated approval in the language of the Indian drug controllers. So if you are interested in looking at the regulatory approvals for the vaccines, you can log on to this website. At this point, these vaccines cannot be marketed because they have an accelerated approval at this point. So only the government can buy them, the government can keep a track of them and vaccinate them. Thank you. Any questions? I'll stop sharing the screen. Any questions? We can take the questions.
Thank you, Dr. Dinesh. That was very well outlined for all of us, and a lot of gray areas have been cleared about the platforms, mRNA or the two adenoviruses as the Sputnik is there. A lot of things you have clarified. Any questions from the postgraduates or the consultants, please? Any questions? Dr. Dinesh, can I ask you one question, please? My query is about these adverse reactions and these Guillain-Barré and transverse myelitis. Though it's rare, but it is very disabling kind of a thing. So is there any way in which the future, I'm not saying present, the future vaccine development will go in any way to mitigate these side effects?
Yeah, I think as theoretically, if you are using these wholesale vaccines, so it is there are more chances of developing these adverse reactions with the wholesale vaccines. But these are more immunogenic. You generate a very good immune response to these wholesale vaccines, like the DPT vaccine, like the rabies vaccine. We have like people can get Guillain-Barré syndrome with getting rabies vaccines, but that is just 1 in 1 million is the incidence of getting GBS with influenza vaccines. See, we don't have, this is just an experimental data, but I think theoretically these newer vectored vaccines, the adenoviral vector, the messenger RNA vaccines should have relatively less frequency of adverse reactions, except the Pfizer's vaccines. The Moderna vaccine has shown to a good safety profile. So I don't think, and no way we can find out who gets severe adverse reaction, a life-threatening anaphylaxis. So that is, anybody can have it.
Right. Because what I understood, the Sputnik has got the two adenovirus
Yes.
vectors there. So it is probably more immunogenic, exciting more immune response, which is what we want. So maybe, we can just target the older population to get this, whereas the younger can be given, the single vector or the
Yes,
the
we will have to the
Oxford.
government. Well, but the government will not. Different. Once they have the stock, only thing is that if they have the logistics is that if you have got 1 dose of Covishield, you should get 2nd dose of also Covishield. You should not interchange. If you have got 1st dose of Covaxin, you should get 2nd dose of Covaxin also.
Right,
So
because the Bharat Biotech, what I understood from your lecture, is using the traditional model of the whole virus.
Yes, they are using the whole kill virus. They are using the whole virus as such. They have purified the virus, then sort of kill the virus. Then it has been it has been conjugated onto a particular protein for aluminum hydroxide sort of thing, which is make it more immunogenic.
So can you tell us some more some vaccines which are like this, whole virus inactivated, which are
There
already
are, say,
existing?
like DPT, like the DPT vaccine we have. DPT is a typical example of wholesale DPT. The wholesale DPT has aluminum hydroxide as an adjuvant, so that makes it more bulky, and because of increasing the bulk of the vaccine, it is more immunogenic. So the body recognizes it, the T and B cells recognize it, and they generate a good response.
Right. So in other words, we have a historical experience with DPT. So DPT is something like the Bharat Biotech, the whole inactivated. Do we have, what is the historical parallel of this Oxford vaccine which Serum Institute is doing?
We don't have any. There were Ebola vectored vaccines. In fact, the adenoviral technology has come. They were trying to test these adenoviral technologies for the Ebola viruses.
Okay,
So
okay.
there are few Ebola virus vaccines which have been based on this adenoviral technology. So that is this is also adenoviral technology is also a time tested technology, but the messenger RNA is a newer technology. We know very little about this technology.
Yeah, which the one which is being used by
Moderna, yeah, the
Moderna.
Moderna vaccine is using the mRNA vaccine, mRNA technology.
Right, which is a very new thing, or you can say untested waters.
Yes, that's but this is that how 20 years back the hepatitis B vaccine was a plasma derived vaccine. But
Okay.
these days all hepatitis B are recombinant, they are DNA recombinant vaccines.
Okay,
So things
okay.
will change.
Things will change, but as of now, if you were to, if you somebody was to ask you this question, that which vaccine I should take,
Yes, I would still, if I were to take it,
yeah,
I
if you
would
were to take,
possibly
which
take a
vaccine will you take?
vaccine which is safe and which is less immunogenic, more of a seroprotective response. I would still vouch doing for a Moderna vaccines, or second for an Oxford Zeneca's vaccine. Third, maybe using Bharat Biotech. Fourth, maybe Pfizer's vaccine. This is how I would look at. But
Okay,
the
right.
healthcare authorities have to look at a different
Yeah,
perspective. They
yeah,
have
of
to look
course.
at the
Nee,
cost
nee, that
mainly.
you are right. But I'm just saying, from the technique used to manufacture the vaccine and your experience as someone who has been given vaccines all your life and seeing the response, I just asked this question that, the best would be your vaccine, which you said as very rightly, the Moderna, which is a messenger RNA, but there is
Yeah,
no
this is a
historical
newer
parallel
technology.
with it.
Yeah, this is a newer thing.
Yeah, absolutely. Just
So
kid
may
on
I
the block.
ask something, Dr.
Sure, sure,
Dinesh?
sure.
See, now there is a concern regarding messenger RNA. They feel that we have heard that this messenger RNA can cause change in the chromosomes or genetic alteration. It can done. It does. This messenger RNA. Maybe it's a myth, but we want to clarify that when we use messenger RNA, it causes some changes in the genetic application of the patient, and that is why they are very apprehensive that they will not take the vaccine. Is it true? Because once we know that, just like HPV DNA infection, that infection is carried through
Not
messenger
true.
RNA, and then it causes changes in it gets integrated into the cells.
Yeah,
So-
that's what I have shown you. The if unfortunately the first, I can't go back to the presentation. What they say, the theoretical basis of the messenger RNA is that it gets into the cytosol, the cell. It directs the
It
cellular
integrates into the
machinery to
cell
form the
system.
proteins, and within 24 hours this messenger RNA, the viral messenger RNA, the vaccine messenger RNA gets destroyed in the cell. So it does not get incorporated in the human genome. Okay. So even if it gets involved in the human genome, finally the body has the capability to make repairs in the DNA. We have an enzyme called a DNA ligase, DNA topoisomerase 2. So that is a clipper in the cellular machinery in the DNA, which clips the defective DNA and repairs the DNA. It's a DNA repairing thing. So body has an inherent lot of mechanisms to take care of these. I think theoretically it should, what the vaccine biologists say, that it should not, but it is only time which will tell us.
But Dr. Dinesh, someone who
Second
has an
thing-
autoimmune disorder or a motor neuron disease,
Yes,
already,
possibly they should not, they may
they
not,
should
they should
not
be
take
given
this.
a kill vaccine.
Yes, they should. Then they should
Second
take
question,
which
may
one?
I ask?
Which vaccine should they
Another-
take? Someone who has autoimmune disease.
Another myth that there is, Dr. Dinesh, that nanoparticles is being used to manufacture these vaccines. Now, these nanoparticles can be modulated in a computerized manner from outside. And if these nanoparticles are taken into the system as a vaccine, maybe our data can be accessed by some external uh, technologies or something. Some external people can take away the data, whatever we have.
Yeah,
Is this a
that's
myth?
right, but I'm not really, and unfortunately, see,
No,
there is a lot
these
of,
are myths that we should know.
Unfortunately, as medical scientists, as clinicians, we know very little about the understanding of the what happens in a molecular biology lab. So if you see a molecular biology lab and if you collaborate with these guys, they will help you better. So fortunately or unfortunately, in this epidemic, we have seen a lot of biotechnologists, a lot of drug manufacturers, a lot of people coming together to formulate newer things. So I have been interacting with different people who are making not the conventional ELISA kits, but newer, very fancy things, which I had not really learned till now. So we know a little bit about the biotechnology. So I don't think I'm an expert at this point to tell you what will happen or
I think this
how
is
they will.
more of science fiction,
Yeah,
Mala.
yeah, this is more
Yeah,
of
we know
science
this is
fiction.
more of
Yeah,
science fiction, but then
yeah, that's
But
there
it's,
are
good
it's worth
realities.
question.
trying to look at, but
Good
we
question.
don't know
No,
what will
good.
happen 20 years down the lane, 10 years down the
Exactly.
lane.
Not 20 years, Dinesh. I can say maybe 20 months down the line. Things
Yeah.
are happening
Yeah,
so
yeah.
fast.
See,
But
we never knew what was Zoom. We never
yes,
knew what was Zoom 2 years back.
exactly. So coming back to my question, Dinesh, person with motor neuron disease, which vaccine should they take?
I think the kill vaccine would be a better option. I would
The
say kill
the
vaccine,
Oxford
though. Yeah,
Oxford vaccine.
yeah, either that's also a vectored vaccine. The Bharat Biotech's vaccine would be a better bet.
Bharat Biotech. Yes.
These adenoviral vector vaccines are also non-replicating vaccines. They
Right,
don't
right.
replicate in the human body. Okay,
Exactly.
so there are replicating vector vaccines and non-replicating vector. These are non-replicating vector vaccines, so they are also safe. I will have to still check the details. I'm not in. I don't think I am an authority on this sort of thing, but
Right.
my hunch says that these people with neurological disease should preferably avoid taking at this point of time the Moderna vaccine.
mRNA based
Yeah,
vaccines.
mRNA based vaccine.
Right. Thank you, Dr. Dinesh. Any other
And what's the
questions?
response? What's the response to the question on whether Covaxin can be administered to immune suppressed patients?
Covaxin is
The
the
attenuated.
Bharat Biotech's, the kill vaccine. Yes,
Yeah, the attenuated, the live attenuated. Yeah.
now, Covaxin is not the live attenuated. Covaxin is a kill vaccine. It is a SARS-CoV derived vaccine from the cells, but it is a kill vaccine. They have killed the viruses. It's just their protein component, the antigenic component. It is not a live vaccine. So all immune compromised individuals can safely take this vaccine.
And what about pregnant
Covaxin.
women? Pregnant women?
Pregnant, we don't have, see, unfortunately we don't have a data on the none of the pregnant women were given these vaccines, so at this point of time they are excluded. So but they can take, I think they can take a kill vaccine.
Theoretically speaking, Dinesh, we
Yes,
can take a
theoretically
killed vaccine.
speaking, but in all trials they have been excluded. Children and pregnant women have been excluded.
So I think the next step would be to check them.
Immunocompromised have also been, in diabetics, the hypertensive immunocompromised people were taken in the AstraZeneca's trial, in the Covaxin trial, they have been taken.
Then we should know the data, no? Whether it's effective in immunocompromised people.
Yes, see that what I mean by immunocompromised people is that what really is the people who are oncology patients, the who are on having a primary immunodeficiency, they have not been taken. It is just
Yeah.
mild
But the autoimmune,
immunocompromised diabetics, hypertensives, or who are on certain medications, they have been taken, but not the oncology patients who have been taken
right,
in.
right.
They were
But what about
excluded in the trials.
right? But what about people with autoimmune diseases like rheumatoid arthritis and everything?
I don't think they have been taken in the. I need to see I don't think they have been. They have been excluded in most of the trials.
Even I think the HIV patients have been excluded from this vaccine trial.
Yeah, definitely. HIV has been a criteria to exclude. That is most of the time. Whenever first, at least for the phase 1, phase 2 trials, they exclude immunocompromised people.
Do we have to fill any? Do we have to fill any form where we have to declare whether whatever problems we have?
No,
before
you don't
getting-
have to. You don't have to. The healthcare individuals do. It is a. You don't have to give a consent. It is a self-implied consent. Only the hospital has given over names to the drug authority, to the healthcare authorities, and that data, the mobile phone, and the names are stored in their system on the
So
COVID
that is all-
application.
No, that is
So
all right. But
it is all,
how,
yeah, you don't have to, you have to just, it's an implied consent for everybody.
no, but then how will any other, you see, layman know whether the person can take the vaccine or not?
No,
I mean,
at this point, we don't, it is not for lay people. It is only for healthcare people. It is for frontline workers. So their all names are registered in the COVID system. They will get the SMS on their mobile phones that
Right.
they are due
Right.
for
But
vaccination
Dr.
on this
Dinesh,
and this
no,
date.
no, no.
Just a minute,
Dr.
just a
Dinesh,
minute.
what she's trying to ask is any absolute contraindication for this vaccine. Like
No,
I am
no,
a doctor,
no.
I may be suffering from
Yeah,
something.
no, that's right. Yeah, that is what I was coming to. Even if it's a doctor and the doctor wants the vaccine, so and then the doctor is taking the risk by taking the vaccine, or there's any performer there where you have to
No,
tick
you
that you
don't
cannot
have
do.
to, you don't have to fill up any pro forma. There is no informed consent. This is,
So the doctor is at risk if the doctor is choosing to take the vaccine.
well,
Is it true?
yes, see, this is an implied consent. As I said, this is an informed, implied consent. Once he is walking into the room, in the vaccination
He's
room,
willing for the vaccine. That's it.
yeah, that's how the government has planned it. They are not taking any informed consent on paper.
Right.
See, but Dinesh,
that is
supposing
more
what is the
screening.
absolute contraindication? Supposing I am a doctor, my name comes, SMS comes to me. Supposing I have motor neuron disease, should I
Then
walk into that
well,
center?
this is an, if you get an, this is an, if you are getting an adenoviral vector vaccine, you are still safe. This or a kill vaccine, you are safe. I don't think that this is the Moderna vaccine. It may have a theoretical risk, okay, but I don't think it is going. Even people with neurological disorders can get the vaccines. In fact, they should be getting at least the kill vaccines. So it depends upon, it is an, everybody will have to make a choice for his
That is right.
own
One more
self.
question, Dr. Kaul. When they were doing the trials, did they segregate the immunodeficient people or they did it on everyone? So by that
No,
we will
I
know
don't
the
think
data.
they have segregated those who were, as I said, those who were on oncological medicines, on biologicals. They have been excluded in the trials. They have
But,
not participated in the trials.
but
Only
do I believe that
diabetics, hypertensives, you may not really call them immunodeficient. They have
no, that is,
participated
they
in
are
the
not
trials.
immunodeficient?
They are
Yeah,
not
no,
immunodeficient.
yeah, they are not immunodeficient. No, what I'm trying to say is that in trials, we do have to give them an informed, proper information about the vaccine. So do we tell them that you should not be coming for the trial if you are immunocompromised? Or we are not
Well,
telling
I
them?
don't think that we should go with that message. Only see, as doctors, everybody has his own sense
No,
to
that is all right.
decide what is good for him.
No, what I'm trying to come to a conclusion is if they have not segregated, then we can find out the data of immunocompromised people.
No, they have not. In fact, no, none of the serious immunocompromised people have been taken. They are not taken as volunteers for a vaccine, phase 1 and 2 or 3 studies.
Right.
Only healthy volunteers are taken.
Right. Thank you.
Yeah,
Sir,
Dr.
two
Kaul,
very quick
just
questions, please. I just have two very quick questions. One is, have there been trials done on pregnant women? And second, have there been trials done on children?
There
And
are,
do you have
in
any
the
data?
Covaxin trial, there were few children from 12 to 18 years, but none of the trials, at least the Covaxin trial, did not have children below 12 years. I don't think any of the trial had pregnant women. Pregnant
Okay.
women have been
Thank you.
excluded.
Thank you.
Sir, hello, sir.
yeah,
Yeah.
Dr. Kaul, just one question I have. It is a bit theoretical. Can I just ask?
Sure. If
Yeah,
the organizers permit.
Yes,
just-
Mamta, go ahead.
Yeah. The question is a bit theoretical that after we receive our vaccination, how do we assess our immunogenic status in terms of antibody formation? Of course, we would like to know whether it has produced effect or not. So, what is the all we go by?
Well, as a public health strategy, the government does. The regulatory authorities who are giving the vaccine, they don't want us to do any immunogenic studies. They just want every healthcare individual to take 2 doses of the Covaxin or Covishield, whatever is available, at 28 days interval. But in your individual capacity, if you are interested, after 15 to 1 month after the second dose, you can check your total antibodies can be checked, and later after 3 months you can check the IgG antibodies. In fact, we are going to work with one of the postgraduates to look at the immunogenic response in COVID convalescent patients.
Yeah, basically, we need to assess the immunogenic response.
That can
once
be
we
done
get
we can do it. Any lab can do it. We are doing it most of the time. If we, before giving plasma to the patients, to check whether they have formed antibodies against the virus.
Okay.
Okay.
And
Thank you, Dr. Dinesh. Yes, Mamta.
And if this, what will be the cost of this Covishield, which we are getting in terms of rupees, if we convert 2-3 USD, you said, I think.
Yeah,
in the beginning.
the government was trying to negotiate with AstraZeneca. They have come to with an agreement with them that they are going to supply the vaccine at the rate of 220 rupees per dose to the government. The Serum Institute of India is going to 20 rupees. But once it comes into the market, it may cost around 1,000 to 1,500 rupees.
Okay. Thank you, Dr. Dinesh, for this information. Thank you.
Good afternoon, sir. I just want to ask one thing.
Yeah.
Sir, do we need to repeat the vaccine yearly or something like that?
No, we don't have any data on that. Just the 2 doses are enough. They are going to give about a year's time of immunity. Hopefully, by that, the epidemic should be gone. We hope.
Okay.
So it is not. That's what we are trying to look. I'm trying to look with we're trying to look at one of the postgraduates to see at least the immunogenic response, seroprevalence, sero response for the vaccines.
Sir,
and the disease.
one more thing. You said that Covaxin is targeting the TH cell responses. So
Yes.
that any spike changes or any genetic modifications in the virus, that can be taken care by the Covaxin, because
Yes, that is the thing, because it's a whole cell kill vaccine. So even if there are certain mutations in the virus, that is a theoretical. It's only time which will tell us.
And what about the Covishield?
See, as far whatever we had information from only the mutations are with the UK mutation, which has come, and the South African mutation, which has come as a sort of a new thing for us. But we don't know how many of these people. It is only time which will tell us that who get the vaccine and subsequently get infected in spite of getting the vaccine. So it's a prospective study for us, which time will tell us. It is just a theoretical assumptions we are making at this point of time.
Okay.
Nothing
Thank
concrete and discrete.
Okay. Thank you so much, sir. Thank you.
Okay. I think we.
Yeah, thank you, Dr. Dinesh. That was a
Thanks a
wonderful
lot. Thanks a lot. Thank
explanation.
you for
Thank you
giving me
so
an
much.
opportunity to speak on this platform. Thank you.
Thank you. Thank you, Dinesh.
Thanks a lot.
Yeah, thank you. Thank you,